Now that we better understand the process of translation, I think it's a great time to watch a really high-budget animation that's found on the Virtual Cell website. And you can see the URL there if you'd like to go visit the Virtual Cell website. There's a lot of way cool animations that you may want to also watch, but we're going to watch the one for translation. So let's see if we can see some of the processes that we've already talked about.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly-A tail.
This is in eukaryotic cells, and we'll talk about the poly-A tail in a moment.
mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end.
Also unique
Translation
to eukaryotes.
initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon.
And in prokaryotes, that would be N-formyl methionine.
The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid. The first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Somehow I feel like that video really captures the elegance of translation, and it also provides a really marvelous segue into our next topic, which is to contrast just a bit eukaryotic gene expression from that we've seen in prokaryotes. And certainly one thing that we saw in that video is that in eukaryotes there's some post-transcriptional modifications on the mRNA. You notice the long poly-A tail, which is very unusual to see in prokaryotes, but is post-transcriptional modification that we see generally in eukaryotes. You also notice the methylated cap, and that's something we also see in eukaryotes. In fact, that cap is absolutely unique to eukaryotes. So there are a couple of other things that are different within eukaryotes. One of those things is that eukaryotes have more than one RNA polymerase enzyme. So whereas one guy does the full job in prokaryotes, in eukaryotes we see 3 different RNA polymerases, and we can actually jot them down as RNA pol 1, 2, and 3, very creatively named. So RNA polymerase 1, RNA polymerase 2, and 3. And they each transcribe different types of genes. So RNA polymerase 2 will transcribe those genes that will give rise to mRNAs. And RNA polymerase 1 gives rise to those rRNAs that do their job as rRNAs as a part of the ribosome. And then RNA polymerase 3 is going to get transcribed to give tRNAs and some other small RNAs. So right off the bat, we see that little bit of added complexity within the eukaryotic world as compared to the prokaryotic world. There's another thing that is different, although it's both the same and different, and that is that the promoter sequences in eukaryotes vary a little bit. They still have the TATA box. We see that TATA box, but it's in a different location. Rather than being found coordinated around the -10 region, we find it around the -30 region. So there's a -30 TATA box. And then there are some other consensus sequences at different places. There's a -50 sequence that is a GC sequence. And there's a -100 sequence as well that's a CAT box, a CAAT. So that just is a little bit different than those consensus sequences that we have talked so much about in prokaryotes. So let's jot down, I guess, the fill in the blanks, so to speak, on this section. And just note that there are differences in the promoter sequences of eukaryotes. We already talked about a little bit, but need to write down that the eukaryotic mRNA is modified after transcription, so it does have that long poly A tail. And you might think about the role that that poly A tail may play in protecting the bulk of the RNA transcript from degradation. If an exonuclease is chewing on the end, on the 3' end of a transcript, it's going to have to chew and chew and chew and chew. And I picture a Pac-Man kind of chewing on that 3' end, and it's going to get rather full before it would even hit any coding sequence in there. So polyadenylation can really serve the purpose of protecting the mRNA transcript. The 5' cap is a methylated derivative of guanine, and that is absolutely unique to eukaryotic cells. Polyadenylation is sometimes seen in certain cyanobacteria and other prokaryotes, but it's much rarer to see that in prokaryotes than to see it in eukaryotes. So approximately 200 adenines kind of protect the transcript there. And there's more than one function of these modifications. We also recognize that these may play a role in enhancing the process of translation. So the 5' cap may actually be something that helps enhance ribosome binding and get the process of translation kicked off. So they certainly do play a very functional role within the eukaryotic cell as well. So modifications likely help to stabilize the transcript and probably to also enhance translation. So that kind of gives us an idea of some of the differences there. Another one that I have a feeling most of you have heard of before is that in eukaryotic transcripts we also tend to see introns and exons. So that is to say we tend to see both coding and non-coding regions of the RNA, and those regions are, well, they're termed introns and exons. The introns are the so-called non-coding regions, the exons, the coding regions. So we'll go ahead and move along and write that down. This is also very rare to see in prokaryotic cells. We usually don't see it except for in a few exceptions. There are some archaea and there are some cyanobacteria that have introns and exons, but for the most part this is something that tends to typify eukaryotic cells. But what we recognize is that introns have to be cut out before we can get the full mature mRNA that will be translated, and the exons are what are the coding regions. So both introns and exons are transcribed to give the precursor mRNA, but then in order to give the mature mRNA, the introns are actually cleaved out. And there's actually a whole big complex called the spliceosome that's going to come through and cleave out those introns. So introns are removed by splicing, and there are little sort of signal sequences to either side of an intron. But these are what signal the spliceosome to splice it out. So the spliceosome is a larger complex, but inside of the spliceosome is one of my very favorite things called SNRPs. And SNRPs stand for small nuclear ribonucleoproteins. So I try to write that down. Small nuclear ribonucleoproteins. And I just love SNRPs. So I tell you what, if you guys write SNRPs and tell me what they do on the front of any exam from now on, I will give you extra credit because SNRPs are just way cool. So they're part of the spliceosome that come through and literally cleave out these introns and remove them, leaving only the coding regions. So we end up with a mature mRNA that has only the exon or coding regions. So down at the bottom then of course is that mature mRNA with its 5' cap and its poly A tail. That's basically what we were seeing in that wicked cool video that we just watched. So differences in the eukaryotic system, right? More than one RNA polymerase. We also see the presence of those modifications, the 5' cap and the poly A tail. And finally, we see the presence of both coding and non-coding sequences within the mRNA. And those, of course, those non-coding sequences have to be spliced out. I should make a note, actually, that it was long thought that introns were like quote-unquote junk DNA, meaning that they really had absolutely no purpose at all. But we now understand that there may be some role that introns play, maybe in enhancing expression of coding regions, and certainly no longer entirely thought to be junk DNA. So as we move on from here, we're going to go into a really fun section on gene regulation. So let's go ahead and move on and talk about the regulation of genes, which is something that just makes a lot of sense because we know that genes there's a lot of energy in making gene products. Remember how much energy is put into all of those processes. And so it doesn't make a lot of sense to make gene products that are not needed at all by the cell. For example, a good analogy would be if you're going to the North Pole, you don't really want to pack your bikini. Likewise, if you're, headed to Hawaii, you're probably not going to spend a lot of time knitting wool stocking caps. It would be kind of a waste of time. And the same thing is true of genes. It would be silly to make gene products that are really not needed by the cell. So that means that genes are only turned on when the products are needed. And not all genes are regulated like that, but many of them are. So we can say some genes are in fact always turned on, right? We know people like that. These are called constitutively expressed genes. They're always turned on. These are often housekeeping genes. These are things that are needed by the cell all of the time. They're protein products maybe that form ribosomes or things that are always going to be in action in the cell at any given time. Those will be constitutively expressed genes. But those genes whose products are not always needed will be either inducible by certain conditions or they'll be repressed by certain conditions. So we can have the sort of opposite where there are some genes that are usually turned off, right? Again, we know people like that. But if there's just the right circumstance, they can actually be induced. So there's certain environmental conditions that can allow for that to happen. Now we can start to think about our plasmid that we worked with in lab, and that plasmid is a good example of containing a gene that is inducible. Remember, the GFP gene was only induced when arabinose was present in the culture media. So that was a good example of induction. Some genes are the opposite, where they're generally mostly turned on, but there are some cases where they can be turned off or repressed. Let's just jot down our plasmid as a little bit of a memory device for an example of induction versus constitutive expression. And remember that piglo had a little bit of both going on. It had some constitutively expressed genes, and it had some that were, or it had one that was inducible. So there's the OREC that we talked about, the origin of replication. And then we had the ARIC gene. and that era C gene was constitutively expressed. So this gene was always turned on. So too was the beta lactamase encoding gene. So that gene that we called BLA that encodes for the beta lactamase enzyme was always expressed or was constitutive. Now the GFP gene was the one that was unique in being inducible. So the GFP gene was only turned on by that rare circumstance of having arabinose present in the culture medium. So we could label these. This one was inducible, whereas ARA-C and the beta-lactamase expressing gene were constitutive, always turned on. So I'll just draw an arrow that so too is the ARA-C gene. So good examples of both inducible number 2 and constitutive number 1. We'll see some other examples where we see repression, where genes are generally turned on, but they can be repressed by certain conditions. So let's go ahead and talk about that. The 3 ways in which genes can be regulated, and one of these we know and love very well. We've already talked quite a bit about promoter strength, right? In a prokaryotic cell, the closer the promoter resembles ideal, the more frequently it's going to get transcribed. So remember that sequence of TT-GACA and TATA-AT the -35 and the -10, and those 2 sequences, the closer they are to ideal, would mean the frequency of downstream transcription would be much higher. So promoter sequence is always going to make a difference. And just to give ourselves a little bit of practice on that, let's remind ourselves TT-GACA, so that would have been a G there and a C there, and TATA-AT. So we've got 4 differences from ideal there. So this may be not the strongest prokaryotic promoter because we're definitely seeing some differences. Let's look here. TT-GACA, there's just one. TATA-AT, ooh, definitely a stronger promoter. I bet we won't find a stronger one. TT-GACA, right, that one's already weaker. And then TT-GACA, TATA-AT. Okay, so our strongest promoter is in fact that second one. So we know that promoter sequence is one of the things that just regulates gene expression. The second way that genes can be regulated is when proteins literally bind to DNA and get in the way of the RNA polymerase enzyme stopping transcription, or vice versa. They can actually bind to and make the promoter more accessible to RNA polymerase, actually enhancing transcription. So this is what we call modulation by DNA binding proteins. Modulation meaning either, repression or activation of expression of those genes. So this is way number 2 in which genes can be regulated by DNA binding proteins that either do one of 2 things. They either block, that is repress, or they enhance, that is to activate. So we can look at 2 different kinds of DNA binding proteins. One that we literally call repressors that bind and block and get in the way, and the second that we literally called activators that bind and enhance expression by making the promoter more accessible to the RNA polymerase enzyme. So let's go through and look at those two categories. We'll just go ahead and list those 2 categories. Repressors are the first, and of course repressing, they're exerting negative regulation. They're like the they're like the jackknife semi truck on I-80, right? They're literally stopping the whole works. They're getting in the way and exerting negative control. So these are going to bind to a site downstream of the promoter where they get in the way and block transcription. This particular sequence is called an operator. So in this picture, we can better make out what's going on. The promoter, where of course the RNA polymerase binds and recognizes, but then downstream of that is this operator sequence. And that's where a repressor is going to bind and literally physically get in the way of expression of any of the genes that might be downstream. So it's going to block RNA polymerase from passing that point. I think that's pretty easy to visualize, right? But the opposite is a little bit harder to visualize. visualize, that is activators that enhance or exert positive control on a gene construct. These are going to bind to a different site and facilitate the transcription. Now, you can think about it this way, right? Literally bending the DNA so that the promoter will become more accessible to RNA polymerase. And that's a lot of what these activators will do. Rather than binding downstream of the promoter, they generally bind upstream of the promoter at a site that we call the activator binding site. Very fittingly named, right? And it actually leads to the enhancement of expression of any downstream genes. So this makes a lot of sense, right? Repressors repress, activators activate, like everything is, hunky-dory, right? But the problem is that things get a little bit more complex here. That is to say that just as enzymes were regulated, and we talked so much about the ways in which enzymes could be regulated, both repressors and activators can also be regulated. Oh boy. So what we're saying is that repressors and activators are allosteric proteins. That is, there are effector molecules that can bind to and regulate whether repressors repress or whether activators activate. For some reason, I was thinking back to Pete's exam, and he did a great job of describing allostery and how we could get either inhibition by effector molecules or activation by effector molecules. And that's true of DNA binding proteins as well. So they too, like enzymes, can be allosteric, and the binding of effector molecules can alter their ability to bind the DNA. It's analogous, really, to enzymes where they were binding substrate, right? Enzymes were binding substrates and their effector molecules changed their ability to bind their substrates. Well, in the world of DNA binding proteins, the effector molecules bind to those proteins and affect their ability to bind DNA. So they may either induce allow the binding, or they may stop the binding, and we're going to talk about sort of the lingo that goes behind some of that. We'll begin with repressors and looking at how repressors can be allosteric proteins. The first mechanism that we'll look at is when a repressor alone can't repress. That is, a repressor alone won't bind to the operator, but requires an effector molecule that enables it to bind. So jotting that down, the repressor alone cannot bind to the operator, but when the effector molecule finds and binds to that repressor, it does allow it to repress. So this is kind of confusing because in this case the effector molecule is enabling the repressor to repress. So we call it a co-repressor because of that. So the effector here is termed a co-repressor. It enables a repressor to repress. That's a mouthful, but it does enhance the ability of that repressor to block transcription. So finish writing so that I can show you cheesy animation number 543. No, probably not at that many, but this is kind of a fun way to show what's going on here, that the repressor alone will not bind the operator. So there's the repressor and denied, right? So we recognize alone it's not going to get in the way of transcription. But once a co-repressor is present, shown here in the red box, it binds to the repressor just as effectors do. It changes its conformation and a sort of domino effect of amino acid transitions that lead to then the conformational change of the repressor, enabling it now to bind to and repress. So repressor plus co-repressor yields a repressor that can repress. So here's one way in which we see allostery exhibited. A second way, we'll call mechanism 2, is when the repressor alone does repress and get in the way, but under certain circumstances that repression is induced. So in this case we call the effector molecule an inducer because it's taking what was repressing and keeping it from repressing. Well, so you're thinking, thinking, well, why is that called an inducer? Isn't it stopping the binding? It's called an inducer because if a repressor is not repressing, it induces the expression of the downstream genes, right? So it stops the repressor from getting in the way of the downstream genes. So we're always referring to whether or not the downstream genes are turned on or not. And in the case of the repressor repressing, they're not, but when the inducer binds, it allows expression of those downstream genes. So we call this effector molecule an inducer. So yet another cheesy cartoon where the repressor alone does bind to the operator, but then in the presence of an inducer, it is now denied. Okay, so these are the two mechanisms, and I want to end today by looking at how those two mechanisms, a couple of examples of those two mechanisms. We'll begin by looking at the TRP operon, which is the operon that allows for expression of genes that synthesize and build tryptophan. It's a 5-gene operon, so we're going to go ahead and jot it down, write it out, showing the coding and template strands of DNA. And on this construct, we have, of course, the promoter, the operator, and then finally downstream we have genes 1, 2, 3, 4, and 5. And if all of these gene products are made, then tryptophan gets built. So what that means is, let's write this down, genes that encode for tryptophan biosynthesis. So recognizing that, we might say to ourselves that if tryptophan is needed, then those genes are on. If tryptophan is not needed, then those genes should be turned off. So we can see this coming, can't we? Tryptophan is going to be the co-repressor of the repressor. So what will typically happen is that the repressor is unable to bind to the operator. But this is a case where the effector molecule tryptophan is a co-repressor. So it's going to come and bind to, in high quantities, it will bind to the repressor and say, whoa, there's plenty enough of me, right? And literally enact its own repression of the genes that make itself. So it will act as the co-repressor. So let's write like this. So repressor plus tryptophan then forms something that can now repress. I'll just show tryptophan as a little blue molecule there. And now we'll recognize that this repressor is capable of binding to the operator and blocking expression because the genes are no longer needed. There's plenty of tryptophan, no need to synthesize more. So here we're seeing mechanism 1 in action where the effector molecule is acting as a co-repressor. So let's jot that down. On the TRP operon, it encodes for 5 enzymes that catalyze tryptophan synthesis, and tryptophan itself serves as a co-repressor. Mechanism 2 is the opposite, where the effector molecule serves as an inducer. And a good operon to study that on is the lac operon. So on that operon, rather than 5 genes, there are 3. But likewise, there is an operator. So downstream of the promoter, we see an operator that I'm just labeling O, and then 3 downstream genes. These 3 genes encode for enzymes that degrade lactose. So that means that they only need to be made in an environment where lactose is present. So that starts to make sense, doesn't it? That would mean that lactose will be the inducer of the repressor. So in this case, the repressor generally binds and blocks at the operator. But if lactose is present, and it's actually a derivative of lactose that does this, it will bind to the repressor and it will induce expression of the genes. So it'll stop that repressor from repressing. So we're going to get a change in conformation once the lactose is bound. And now that will no longer be able to bind the operator denied, right? So this is a good example of mechanism 2, where the effector molecule is an inducer rather than a co-repressor. And we're soon going to see that expression of the lac operon is even more complex than what we're drawing here, but this is a good start in understanding it, and certainly in understanding mechanism 2, where we see that the expression of these genes to make the enzymes that degrade lactose is indeed controlled by the presence of lactose, where it acts as an inducer. So that will then allow for expression of genes 1, 2, and 3 on the lac operon. We're going to stop there for today, and you can look forward next time to talking about even more complex gene regulation, right? Hard to believe that it gets even cooler, but it definitely does.